In this video, we'll look at the biological inspiration behind neural networks. So we've seen a mathematical definition of what we mean by a multi-layer neural network. Mathematically, we have an input vector, which we call the input layer, and then this vector is transformed into hidden layers, where each hidden layer is composed of hidden units, which are all computed by a linear transformation followed by nonlinearity. And then so we compose these operations until we reach an output layer, which makes a particular prediction. If we're doing classification, it tries to predict the class to which the input belongs. So now we'll look at what is the biological inspiration behind this model. Why is it that we call these little units here neurons? And how did this mathematical formulation, how was it derived from an inspiration from actual neurons and the actual brain? Let's first talk about this idea of structuring a model into several layers of pre-processing of hidden units. One inspiration that we often refer to is from the visual cortex in the brain. That is the part of the brain that interprets the visual stimuli that we get from our retina. So what we know about the visual cortex is that it is decomposed into layers of regions that are connected to each other. So information from the retina is passed to a first region called LGN, and then eventually it reaches another region of neurons, which is called V1. Now, V1 is known to have neurons that are sensitive to particular simple visual forms like edges and corners. And then this information, so the activity of these neurons is influences the activity of another region called V2. And then eventually it reaches another region called V4, which now starts extracting some features related to more intermediate visual forms or features. So essentially groups or combinations of simpler features like edges and corners. And then this eventually reaches PIT and then AIT, where we get neurons that are sensitive to particular faces or particular objects in the visual field. So we see that the processing of information in the visual cortex goes through these layers or regions that are put in sequence into the processing of the brain. Now, we can look at a cartoon explanation of what we can do with such processing of or a sequence of regions. Say we have a first region that might extract different edges in an image, and then these features would be reused by a subsequent region, which would then combine these features. So for instance, we could have a nose detector that would combine this feature with this feature. So this would be this line here and this line here. We could have a mouth neuron, which would detect mouths by detecting the presence of this line here and this line here, which is this line here and this line here. And then something that would detect two eyes, so that would be this feature and this feature. And then again, the activity of these neurons that can detect noses and mouths and eyes could be combined to get a whole face detector, which would be active if the neuron that detected a mouth, a nose, and then two eyes were actually activated, were detecting these particular features. Okay, so this is the brain inspiration behind having several layers of processing of hidden layers. So each of these parts here would correspond essentially to a layer of neuron in our artificial neural network. Now let's talk about individual neurons and the way that mathematically we write them out. Where does that come from? So let's talk a little bit about the actual brain. Now we estimate that there are about 10 to the 10 and 10 to the 11 neurons in the human brain. Each neuron receives information from other neurons through their dendrites. So dendrites is kind of like the ears of the neurons that so it receives the information from other neurons it is connected to, and then information that comes from these dendrites is processed within the cell body of the neuron, which is called the soma. Then the neuron will send information through a cable, quote unquote, which is called the axon. And then this axon is going to be connected with other neurons' dendrites, and the point of connection between a neuron's axon and the branches out of its axon and the other neuron's dendrites is going to be called the synapses. That's the point of connection where an axon reaches some other neuron's dendrites. There's a cartoon illustration of all of these different terms. So for a given neuron, it has some dendrites. So these are these arrows here. And this is how information is being sent into the cell body or the soma of the neuron. Then there's some chemical activity that happens here. And the result of this chemical activity, this results into an electrical signal that is sent through an axon. And this axon is then becomes some branches which are eventually connected to other neurons' dendrites, and the point of connection between these dendrites, so this little square here in this cartoon illustration, this point of connection would be synapses. So the synapse is where the axon reaches another neuron's dendrites. Now, we'll call an action potential an electrical impulse which travels through a neuron's axon. So this is actually how a neuron will communicate with other neurons. This is how it's going to send information. And so essentially, this action potential corresponds to a spike in the electrical voltage, electrical potential, sorry. So it spikes in voltage in of the axon. And now an action potential that is generated at a neuron if it receives enough of the right pattern of spikes from other neurons that is connected to. And so essentially this level of activity from the other neurons from which a given neuron is receiving information through its dendrites. If this amount of activity reaches a certain threshold and has the right pattern, then the neuron in question will also generate an action potential, will also spike. Now, neurons will actually generate several spikes every second. So there's actually going to be a frequency of spikes, and this is called the firing rate of a neuron. And this is actually, one of the main information or main characterization of the activity of a neuron. So we'll often look at its firing rate as a description, a number that will describe whether a neuron is very active or not. Actually, neurons always fire a little bit, and this sort of constant level of firing is known as the spontaneous firing rate. But they will actually fire much more given the right stimulus, that is, given the right pattern of firing rates in the other neurons it's connected to, then our neuron will might increase its firing rate and might generate many more spikes. Now, so essentially the firing rates of some other neurons that are connected to a given neuron, so we'll call those neurons the input neurons, they will effectively combine to influence the firing rate of the neurons it's connected to. Okay, so if we have one neuron and then it's actually connected to many more neurons, so they have their axon and eventually it reaches some dendrites of this neuron here, the firing rates or information will be propagated in this way. So the firing rate of these neurons will influence the firing rate of that neuron that these neurons are connected to. And so depending on the properties of the dendrite and the axon, so the dendrite of this neuron and the axons of these neurons, a neuron can either work, so one of these neurons can either make the firing rate of that neuron increase or decrease by increasing its own firing rate. So if we say that the firing rate of, say, that neuron will tend to increase the firing rate of that neuron, then we'll say that this neuron here excites this neuron here, and otherwise if it decreases it, then we'll say it will inhibit the firing rate of that neuron. Okay, so now with all that being said, what's the relationship with our artificial neurons? So, well, they try to essentially approximate this process of firing rates that are play together to influence the firing rates of other neurons. So the activation or the output activation of a given neuron, so after the activation function, we can think of it as sort of a firing rate. So this is, if you have a sigmoid, it's a number between 0 and 1, so that's not necessarily the right range for a firing rate, but we should think of the activation as sort of the equivalent of the firing rate of that neuron. Now the weights between the neurons, they actually model whether some given neuron will excite or inhibit other neurons. So if we have a positive connection between two neurons, it means that the input neuron will excite the output neuron, the neuron it's connected to. And if instead it inhibits, so then we'll have a negative connection. Okay. So these weights essentially translate this idea that firing rates can either increase or decrease the firing rates of other neurons. And then the activation function and also the bias within the neuron, they're sort of a model for this thresholded behavior of action potentials. So I said that the firing rates or the firing activities of the neurons that some given neuron is connected to needs to reach a certain level for this neuron to actually spike. And so this is what the activation function and the bias sort of try to model. So if we have a sigmoidal function, it will take this form here. And then the bias will determine what's the basic pre-activation of the neurons. If the bias is negative, then so this would be at 0. If the bias is negative, then the pre-activation would start here. So this is saying that the pre-activation, where we combine the activation of the neurons it's connected to, so they're sort of firing rates. In this particular case, it would mean that it needs to go quite a bit over 0 to reach the maximum firing rate of that neuron. Okay, so we can think of this as a sort of soft threshold, and so this is what the activation function in the bias is trying to model. This a soft version of this thresholded behavior. Okay, so that's all we'll say about the biological connection between artificial neural networks and real biological neurons. Often when we do develop neural networks, we don't always go back and think about, okay, let's try to make our neurons more biologically plausible. Some people use this in their research to improve and design better neural networks, but some other will instead focus on the mathematical properties of artificial neurons and try to play with that. But it's still important to know that there is some, though fairly weak, connection between real neurons and artificial neurons. And actually, if you want to see some neurons in action, I suggest you go look at the recording of the Hubble and Weasel experiment, where a neuron in V1 region of a cat visual cortex is being recorded. So you can actually hear the neuron spike as a visual stimuli, which corresponds to some lighting pattern, is being changed in the visual field of a cat. So this is a nice experiment to watch and to get a more tangible feel of what an actual neuron's behavior looks like.